Welcome back to the online classes of biology. In the last class, we have studied about the autotrophic nutrition. In the autotrophic nutrition, we have studied about that photosynthesis is an process which is helpful for the preparing food material in the autotrophs, that is in all the green plants and a few photosynthetic bacteria. We have already studied photosynthesis absorb the carbon dioxide and it is react with water in the presence of sunlight, and it is produces glucose, releases water and also oxygen. Here we are going to observe that all the factors which are essential for the photosynthesis, and we are also going to prove the factors essential for the photosynthesis with some of the activities. Among them, first of all, we are going to study about the carbon dioxide. We know that carbon dioxide is one of the major gas present in our atmosphere. In our atmosphere, there is a 0.03% of the carbon dioxide is present. Whenever we are going to observe that this carbon dioxide plays a crucial role in the photosynthesis. Here we are going to start our experiment to prove the carbon dioxide is essential for photosynthesis. We know that we already discussed carbon dioxide plays an important role in the photosynthesis. Without carbon dioxide, there is no chance for the photosynthesis. So here we are going to perform an experiment to prove the carbon dioxide is essential for photosynthesis. That is our aim. For this, we are going to collect some of the apparatus. What are the apparatus we are collected for this experiment means, first of all, we are collected one of the healthy potted plant, and we are also collect the wide mouthed glass bottle, and we are also going to be taken iodine, KOH solution and splitted rubber cork and Vaseline or grease. These are the apparatus we are collected for this experiment, and then we are going to move on to the procedure. Before going to start our experiment, first of all, we should keep the entire healthy potted plant in the dark room to destarch the plant. How we can destarch the plant means, whenever we can kept in the dark room, this plant can utilize the starch present in the plant body and it is going to be destarched plant. After 3 to 4 days duration, that means whenever it is destarched, later we are going to be collect this destarched plant. And now we are going to be select any one of the broad leaf from the destarched plant without detaching from the plant. And now we will take one of the wide mouthed glass bottle, and we are going to be pour 5 to 10 ml of potassium hydroxide solution in the wide mouthed glass bottle, and we will close it with the help of the rubber cork, and we are going to be insert the one of the broad leaf into the wide mouthed glass bottle. Here we should insert only half of the leaf into the bottle, and later we should apply the Vaseline or grease at the mouth of the bottle, which is prevent the entry of gases. And then we are going to be kept the entire setup under the sunlight. Whenever we will place the entire setup under the sunlight for a few hours, after few hours of the duration, we should go for the test for starch. Whenever we will observe that test for starch, we will carefully remove the wide mouthed glass bottle, and then we will detach the leaf from the plant. Now we will conduct the starch test. This leaf is taken in one of the petri dish, and we will pour the iodine solution. Whenever we will pour the iodine solution on the leaf, we will go for the observation. In our observation, we observed that half of the leaf present in the bottle remains constant, and half of the leaf present outside of the bottle turns into blue-black color. Why? It is going to be show the differences. Whenever we are going to observe that half of the leaf which is present inside the bottle remains constant, and half of the leaf which is present outside of the bottle turns into blue-black color. Whenever we will observe the difference, we will go for our inference. Whenever we will go for our inference, we should observed that half of the leaf which is present inside the bottle is not having the carbon dioxide, and half of the leaf which is present outside the bottle is present in the atmosphere. So it is absorb the carbon dioxide from the atmosphere. So here we are going to be show the difference. That means whenever we will observe that where the carbon dioxide is available, their photosynthesis is going to be performed by the plant, and it is going to prepare the starch so that it is going to be turns into blue black color, that is outside of the bottle. And inside of the bottle it remains constant because there is no availability of carbon dioxide. So it is unable to perform the photosynthesis, and it is unable to prepare the starch so that it is remains constant. That means here whenever we will pour the KOH solution, this KOH solution is removes the carbon dioxide present inside of the bottle, so that it is not able to perform the photosynthesis, and we will keep the bottle airtight so that it is unable to entry of carbon dioxide into the bottle. So there is no carbon dioxide. That means here completely absence of carbon dioxide. So there is no carbon dioxide, there is no photosynthesis, and there is no preparation of starch, so that it is not turns into blue black color, it remains constant. By this experiment, by this observation, we will prove that carbon dioxide is essential for the photosynthesis. Why? Because where the carbon dioxide is absent, there is no chance for the photosynthesis, and there is no preparation of the starch, and it is not turns into blue black color. So this is an experiment. We are going to be proved the carbon dioxide is essential for photosynthesis. Whenever we are performing this experiment, we will take some of the precautions. What are the precautions we are going to take for this experiment? Means here we will observe that whenever we will keep the leaf into the bottle, it must be away from the KOH solution. Why? Because this KOH solution is alkaline in nature, so it is going to be damage the leaf. So it must be away from the KOH solution. At the same time, the bottle must be airtight. That's why we are going to be close the bottle with rubber cork, and we are going to apply the Vaseline or grease at the mouth of the bottle. Why? Because if the entry of gases will be takes place, there is an changes in the result, so that we must cut the entry of carbon dioxide. Whenever we will cut the carbon dioxide means, that's why we are going to be apply the Vaseline or grease to the mouth of the bottle. So finally, by this experiment we are show that carbon dioxide is essential for photosynthesis. That is an experiment we are performing here. Remember that all the apparatus we are collected here performing experiment, and before going to start the experiment, plant must be de-starched. Why? Because in the starched plant we can't able to show the result. That's why we are going to collect the de-starched plant here. For the de-starching of the plant, we will keep the plant for a few days in a dark room. Whenever we can kept in the dark room, that healthy potted plant turns into de-starched plant. With the de-starched plant only, we are going to start the experiment. That is an experiment to prove the carbon dioxide is essential for photosynthesis. The last topic we have studied about the carbon dioxide is essential for photosynthesis. Now here we are going to be study about the light and photosynthesis. Generally we are going to be studied about that carbon dioxide is essential for photosynthesis. At the same time, light also essential for the photosynthesis. Here, most of the scientists are going to be studied about the importance of light. During the time of the Joseph Priestley, whenever he discovered about the importance of air, on that time there is no one can identify about the importance of the energy and how the energy is going to be released. During that time, there is one of the scientists, his name was John Ingenhousz. In the year 1779, he was started experiments on the hydrilla twigs, and he is going to be observing that oxygen is going to be liberated during the photosynthesis in the presence of sunlight. Whenever oxygen is released, for that also plants requires energy. From where this energy is obtained? On that he is going to be conducting experiments. Later there are so many other scientists are going to be conducting several researches, and finally Engelmann in the year 20th century he is going to be find out he is going to be proved with his experiments. According to that, generally we know that whenever we are going to be observe the equation of photosynthesis, here we can find out that carbon dioxide react with water and produces glucose, water, and releases oxygen. We know that where it is going to be releases oxygen, and when it is going to be releases oxygen means it is releases oxygen in the presence of sunlight. So generally whenever we can observe the importance of sunlight, there is a lot of things are going to be observed here. First of all, here it is going to be releases oxygen, that is we are going to be studying here. Whenever we can conduct an experiment to prove the importance of oxygen, we are going to be performing one of the lab activity. In that lab activity we are going for that our aim. What is our aim here means our aim is to prove that oxygen is liberated during the photosynthesis. For that we are going to collect some of the apparatus like beaker, test tube, glass funnel, and hydrilla twigs. These are the apparatus we are collected for this activity. Whenever we are going to be collecting all these apparatus, let's move on to the procedure. How we are going to be perform the experiment. In this experiment, first of all, we will take one of the large beaker and filled with water up to 3/4. Whenever we can fill it with water, later we will take a glass funnel. This is a glass funnel. Here we are going to be observe that it is a broad portion, that is we get called as the mouth, and this is called stalk. At the mouth of the funnel we will place the few hydrilla twigs. Whenever we will place the hydrilla twigs, later we will collect this glass funnel and place it in the beaker containing water in the inverted position. First of all, here we will take the glass funnel. In the glass funnel mouth we will place the hydrilla twigs, and the entire funnel along with hydrilla twigs is placed in the water containing beaker in the inverted position. Later we will take one of the empty test tube, and that empty test tube is placed on the stalk of the funnel in the inverted position. First, funnel will be placed in the water containing beaker in the inverted position. Later, test tube is also placed on the stalk of the funnel in the inverted position. Now the entire setup will be placed under the sunlight for the few hours. Whenever we will place in the sunlight, later after few hours of duration we have to observe that small air bubbles are arises from the hydrilla twigs. Generally we will observe that air bubbles from the hydrilla twigs. Whenever we will observe the hydrilla twigs, whenever we will observe the air bubbles from the hydrilla twigs, we are going for the collection of gas. How we are going to collect the gas? These air bubbles are coming from the hydrilla twigs, and we are going to be leave it for a few minutes. After the few minutes, slowly we will remove the test tube from the stalk of the funnel. Carefully we will remove the test tube from the stalk of the funnel, and whenever we are going to be coming up to the end of the water, we are close the mouth of the test tube with our thumb finger. During the collection of gas, carefully we will remove the test tube from the stalk of the funnel, and at the end of the water we are close the mouth of the test tube with the help of the thumb finger. This is a collection of gas. Now we are going to be collect the gas, but we don't know which gas it is. Here we are going to prove the oxygen is liberated during photosynthesis. Just we are observed here gas bubbles are arises. That is an which gas? That is we are going to be proved with our another simple test. How we are going to be identify the either that gas is oxygen or any other gas? For that we will go for that test for gas. Which test we are going to be conduct to identify the test for gas? Just we have to take one of the burning matchstick. We will take one of the burning matchstick, and where we are closed with thumb finger at the mouth of the test tube, we are closed with thumb finger. Carefully we will remove the thumb finger and just we have to place the burning splinter at the mouth of the test tube. During that time we will observe what observation we are going to be find here. Whenever we will go for the observation, during that time that burning splinter burns more brightly. It burns more brightly. Whenever it burns more brightly, with that test we are confirmed that it is oxygen. Why? Because we know the properties of gases. If it is any other gas means there is no chance for the burning brightly. Why? Because if it is carbon dioxide, it is going to be puts off the light, puts off the flame. At the same time, if it is hydrogen, we know that it is also puts off the flame with pop sound. But here it burns more brightly, so that we are confirmed that it is an oxygen gas. Now by this experiment we are going to give the conclusion. What conclusion we are going to be given with this experiment means we can say that oxygen is liberated during the photosynthesis. That means with the experiment, with the hydroelectric experiment, we are proved that oxygen is liberated during photosynthesis. That means in the presence of sunlight, plants performs photosynthesis and it is releases oxygen. By this experiment we are proved that oxygen is liberated during photosynthesis. Okay, in the last activity we are studied about that oxygen is liberated during the photosynthesis in the presence of sunlight. In this experiment we are going to be studied about the light is essential for photosynthesis. We know that photosynthesis occurs in the presence of sunlight. Here we are going to be studied about the light. We already studied John Ingenhouse and Engelmann can prove that oxygen is liberated during the photosynthesis in the presence of sunlight only. Sunlight which is called that is an visible light. We know that there is different types of rays are there. Among them we are going to be studied about that visible light. In the visible light it is available in the form of the rays. The light rays are called as the photons, and the energy stored in the photons is called as quanta. The light rays is called photons and the energy stored in the light rays is called as the quanta. Before here the visible light is wavelength is up to 400 to 700 nanometers, and below the 400 nanometers which is we can call as an ultraviolet rays, and above the 700 nanometers which is consisting of the infrared rays. Below 400 ultraviolet rays is there and above 700 nanometers infrared rays are present. Here we are going to be that is an photosynthesis is occurs in the wavelength of 400 to 700 nanometers. That means it occurs only in the presence of visible light. Generally our visible light which is going to be consisting of the different colors. Generally it is appeared colorless or white in color, but which is going to be consisting of the different colors, that is we will call as the VIBGR. That is a violet, indigo, blue, green, yellow, orange, red. These are the different colors are present in the visible light. That is we will call as a VIBGR. and that is an violet, indigo, blue, green, yellow, orange and red. All the rays of light is not suitable for the photosynthesis. Photosynthesis is occurred mostly at the point of the blue and red colored rays only. Generally here green colored light is there. This green colored rays are going to be reflected by the green leaves. Why? Because already leaves are consisting of the chlorophyll. That chlorophyll is also green in color, so that green color is going to be reflected back, and blue color and red colored light rays are this is essential for the photosynthesis, and the maximum rate of photosynthesis will be takes place in the presence of blue and red rays. So here that is an light. Generally we are going to be study about that light. Here how this light is utilized for the photosynthesis and how the photosynthesis will be takes place in the presence of sunlight, that is we are going to be studied, and that is we are going to be conducting and one of the activity. In this activity we are go for that, first of all we will go for our aim. What is our aim here? Our aim is just we have to say that that is an sunlight is essential for photosynthesis. How the sunlight is essential for photosynthesis, that is we are going to be proved with one of the activity. Our aim is also that is to prove the sunlight is essential for photosynthesis. To prove the sunlight is necessary for photosynthesis. For that we are going to be collect the some of the apparatus. What are the apparatus we are collected here means, again we are going to take the one of the healthy potted plant. Healthy potted plant we are going to be collecting here, and we are also collecting one of the light screen, and we will take the iodine solution for the starch test. These are the apparatus we are collected for this activity. And here just we have to take that healthy potted plant, and now we are going to move on to our procedure, how to perform the activity. Here we will take the one of the healthy potted plant. This healthy potted plant, before going to perform the experiment, we are going to make the de-starch. We know that already how to de-starch the healthy potted plant. Just we have to keep the healthy potted plant in the dark room for few days. Within few days this healthy potted plant can utilize the stored starch present in the leaves, and it is becomes starch free plant. That is we will call the starch free plant, or we will call as an de-starched plant. Now we will take this starch free plant, and in this starch free plant we will select any one of the broad leaf, and just we have to attach the light screen cutting with any of the design. What is light screen? Just those are not able to penetrate the light through them, that is we will call as a light screen. Just we have to take any one of the cardboard or paper or film, any one of these we are going to be collecting as a light screen which is not able to penetrate the sunlight through them. Just we have to take one of the cardboard, and that cardboard is cutting in the size of the leaf, and cut it into any of the design in the middle of the cardboard. Just I have to cutting here as a shape of the S, otherwise we will cut the shape of the star, whatever it is. In the shape of any of the shape we are going to be cutting in the middle of the cardboard, and just we have to take that cardboard and placed on the any one of the broad leaf of starch free plant. On the any one of the broad leaf, just we have to place a light screen, and it is going to be hold it with the help of the clamps. Any of the clamps we are going to be taking here. Just we have to place the cardboard on the leaf, and both the sides we have to hold with the help of the clamps, and then we have to place the entire setup under the sunlight for few hours. Here the entirely we will take a starch free plant. just we have to take any one of the broadleaf and we have to cover the leaf with the help of the light screen cutting with any of the design. Now it will place under the sunlight for few hours. After few hours of duration, now we will remove the light screen. Then we have to detach the leaf from the plant. But we have to remove the light screen and detach the leaf from plant. Then we have to go for the starch test. How we are going to conduct the starch test? We know already, we are studied in the last experiments also. Just we have to take the leaf and the petri dish. This leaf we are going to be taken in one of the petri dish. And we have to pour some of the few drops of iodine solution. Just we have to pour the iodine solution with the help of pillar. That is, we have to pour the iodine solution. Whenever we will pour the iodine solution on the leaf, now we have to move on to our observation. What we have to observe, that will be noted down in our observation notebook. What's going on to the leaf whenever we can pour the iodine solution? Just we have to find out that we are observed that whenever we will pour the iodine solution, where the light is penetrates or when the light is passes, there only it is turns into deep, that is a deep blue black, and remaining portion it is constant. Just we have to observe here, where the light is passes, there only it turns into blue black color. Where the light not penetrates, it remains constant. Just we have to see here, whenever we will place the cardboard on the leaf, here the entire portion is covered with cardboard, so there is no chance for the entry of sunlight. Where we will cut in the shape of design, where we will cut the shape of S, there only chances of the entry of sunlight. And both side of the cardboard there is an empty space is there, so there is also chances of the entry of sunlight. That means where we will cut the design and where the cardboard is not there, there only sunlight is able to penetrate, and remaining portion is not able to penetration of sunlight. So that by this observation we are going to give our conclusion. What is our conclusion we are going to given here means, just we have to see here, where the light is available, there only it is going to be performs the photosynthesis, and there only it is going to be prepare the starch, and because of the presence of starch it is turns into blue black color. And where the light is not available, there no chance of the performing photosynthesis, and it is unable to prepare the starch, so it is remains constant. That means where the light penetration happens, there only it is performs photosynthesis, and there only it is prepares the starch, and there only it is turns into blue black color. Where the light is not available, there is no chance for the performing photosynthesis, and there is no chance to prepare starch, and it is remains constant. So that by this experiment we are proved that light is essential for photosynthesis. Why? Because where the light availability is there, there only it is performs photosynthesis, and it is prepares starch, and it is turns into blue black color during the starch test. That means by this activity we will conclude that sunlight is very essential for the photosynthesis. This is an experiment we are conducted to prove the importance of light in the photosynthesis. These are the experiments we are studied related to the photosynthesis. In these topics already we are studied about that carbon dioxide is essential for photosynthesis, sunlight is essential for photosynthesis, and we are studied about the oxygen is liberated during the photosynthesis in the presence of sunlight. All these experiments we are going to be related to the photosynthesis. At the same time, here all these experiments shows that importance of the carbon dioxide and importance of the sunlight. Okay students, this is about the importance of sunlight in the photosynthesis. Once you have, all of you refer the all experiments in the books and you have to practice the experiments. Thank you.